Have you ever contemplated the mystery of life, how we pass traits from one generation to the next? This wonder is not a work of some magical spell, but the result of a complex yet fascinating process known as DNA replication. DNA replication is the biological mechanism that copies the genetic information contained within the DNA of a cell, allowing that information to be passed on to its offspring. It's a critical process for the continuation of life, serving as the basis for biological inheritance. Without DNA replication, life as we know it would cease to exist, but it doesn't stop there. DNA replication also plays a vital role in maintaining our body's equilibrium, or homeostasis, and the integrity of our bodies. This process ensures that each new cell has the same genetic information as the one it originated from. It's the magic of DNA replication that makes the transmission of genetic information possible. So how does this seemingly complex process of DNA replication actually work? Well, let's break down this intricate dance of biology into simpler steps. DNA replication is a process that occurs in every living organism as it's essential for life. It involves the duplication of a DNA molecule into two identical replicas. At the heart of this process are enzymes, the biological catalysts. They spring into action when a cell decides to divide. Among these enzymes, the star of the show is DNA polymerase. This enzyme has the crucial job of adding nucleotides to the new DNA strand, effectively building a duplicate of the original. But before DNA polymerase can work its magic, another enzyme, helicase, steps onto the stage. Helicase unwinds the DNA double helix, separating the two strands and creating what is known as the replication fork. This is where the real action begins. DNA polymerase then takes over, moving along the original or parent DNA strand. As it moves, it adds nucleotides to the new or daughter strand. These nucleotides are the individual units that make up DNA, consisting of a sugar, a phosphate, and a base. The base can be one of four types: adenine, thymine, cytosine, or guanine. The order of these bases determines the genetic code. But there's a catch. DNA polymerase can only add nucleotides in one direction. So while one new strand is built smoothly, the other has to be constructed in fragments, which are later joined together by yet another enzyme, ligase. In this way, the DNA molecule splits and duplicates itself, leading to two identical DNA molecules. Each of these carries the same genetic information as the original molecule, ensuring that when a cell divides, both new cells have the exact instructions they need to function. So, while the process of DNA replication may seem daunting at first, it's really just a matter of enzymes working together to create an exact copy of a DNA molecule. In essence, DNA replication is all about creating an exact copy of a DNA molecule, a process vital for life. But why does understanding DNA replication matter? What are its applications? These are valid questions, and the answers may surprise you. DNA replication is not just a simple biological process. It's the key to transferring genetic information from one generation to the next. This invaluable process allows our bodies to maintain homeostasis and integrity. But it doesn't stop there. The knowledge we have about DNA replication has opened up a whole new world of possibilities. Take genome sequencing, for example. The ability to sequence an entire human genome is now within our reach, all thanks to DNA replication. This technological advance has far-reaching implications, from diagnosing genetic diseases to developing personalized treatment plans. But the applications of DNA replication don't end with genome sequencing. It's also made possible the cloning of genes. Imagine being able to replicate specific genes to study their function, or even to replace faulty ones. This could revolutionize the way we treat genetic diseases. And then there's the breakthrough Cas9/CRISPR technology. This innovative technique, which uses nucleases to cleave a specific portion of DNA and replace it with required nucleotides, relies heavily on DNA replication. It's a prime example of how we can harness the power of DNA replication enzymes to make potential advancements in science and medicine. Another example is the polymerase chain reaction, a technique that uses DNA polymerases to replicate DNA in vitro. This method has numerous applications, from diagnosing diseases to sequencing DNA and developing recombinant DNA technology. Lastly, let's not forget the formation of complementary DNA or cDNA. This process, which relies on DNA replication, has wide-ranging applications in gene expression and protein production. So, the next time you hear about DNA replication, remember that it's not just about copying DNA. It's about unlocking new possibilities, from understanding our genetic makeup to developing cutting-edge treatments for diseases. With DNA replication, we're not just copying DNA, we're unlocking new possibilities in science and medicine. As we continue to unravel the mystery of DNA replication, what does the future hold? The future of DNA replication is a captivating frontier, teeming with potential innovations and breakthroughs. One of the most promising advancements lies in the realm of artificial intelligence and machine learning. These technologies are being harnessed to predict, analyze, and even simulate DNA replication. This can not only speed up research, but also allow for more precise and accurate results. Imagine having an AI that can predict how DNA will replicate under certain conditions or help identify potential errors before they even happen. Moreover, the future of DNA replication could see major advancements in the field of recombinant DNA technology. This technology, which involves combining DNA from different sources, has vast potential in areas like gene therapy and genetic engineering. As we continue to refine our understanding of DNA replication, we can improve the efficiency and accuracy of these processes, opening up new possibilities for treating diseases and enhancing our understanding of genetics. Furthermore, the advancements in DNA replication could revolutionize scientific research. With a more detailed understanding of how DNA replicates, we can delve deeper into the intricacies of life at the molecular level. We could potentially discover new species, explore the genetic basis of complex traits, and even unravel the mysteries of evolution. The potential is truly limitless. However, as we journey into this exciting future, it's important to remember the ethical considerations. With great power comes great responsibility. As we develop the ability to manipulate DNA with increasing precision, we must ensure that these technologies are used responsibly and ethically. As we push the boundaries of what's possible in DNA replication, we are not just advancing science, we are also paving the way for a future where we have a deeper understanding of life itself. This isn't just about understanding how DNA replicates, it's about understanding the very essence of who we are and how we came to be. DNA replication is not just a process, it's a gateway to our future, offering answers to some of life's biggest questions.